Good afternoon. Thank you. My name is Tom Gaborski. I'm the President and Chief Operating Officer of Simpore. Simpore is an early-stage company. It's commercializing a novel membrane technology, really enhances the efficiency of separations, primarily in the life science industry. We're targeting an area that you've heard about several times this morning already, drug development. However, we're not producing drugs. We're creating tools for researchers at the early stage of research, screening, and also there's in vitro assays done prior to clinical trials. Not too long ago, all assays done before clinical trials are done in animals. Today, the majority of those are actually done in cells and cell substrates. All of these assays are done in, you can see, large volumes. This pathway, in general, as was discussed earlier today, in round numbers, costs about $2 billion for a drug that makes it through FDA approval in the United States, and a time span of 10 or more years. A significant amount of that effort is done up front here. On this chart from one of the national organizations suggesting 3 to 6 years. A lot of this in the early stage is done with high throughput screening, and you've probably heard about this technology. It allows people to condense material and assays down, use smaller reagents, allows more work to be done at less cost. However, there's really a big gap. Once the early screening is done in some of these high throughput senses, the preclinical studies, these are the in vitro and animal studies, are really low throughput. There's a significant need here to move high throughput to these in vitro assays. Low volume tools need to be fast and efficient to use fewer reagents. We have the first disruptive membrane technology in decades. A traditional polymeric membrane that's used in the life science industry looks like this on the left. It's a membrane material that is relatively thin. It's microns in thickness, but we're separating materials that are nanometers in thickness. So there's about a 1,000-to-1 aspect ratio between the thickness and the effective pore size. This means there's a significant amount of loss, trapping of material, and it's relatively inefficient. What we've done is we've basically taken the same pore size, but shrunken the thickness of the material to nanometers. So we now have a 1-to-1 aspect ratio of pore size to thickness. This allows much higher efficiency, higher permeability, and most significant, less loss of material. Due to the fact we make this entirely different than a traditional membrane, we also have much better control over the absolute pore size, meaning we can do very precise separations. The markets that we're addressing, as I mentioned, are predominantly in the biopharma space. However, we'll be attacking the research tools initially. These are both in academic and in biopharma industries. For separations, we basically offer the precision of much more complex instrumentation. Traditional membrane filters are great at concentrating, really packing down material, getting rid of excess solutions, maybe changing salt solutions. They're not very effective at separating proteins and other small molecules. These are the exact type of purifications that need to be done at the early stages of biopharma. That work is left up to chromatography and other tools. These tools are relatively expensive, complex, require trained technicians to operate them. Membranes just don't meet those needs. However, our membrane material, because of the precise pore size and the thickness of it, allows some of the precision of chromatography, but with the simplicity and speed of a membrane filter. Likewise, coming down to in vitro assays, the types of cells that are grown in these preclinical studies are oftentimes barrier cell types. Many of the studies that are required by the FDA look at how cells mimic the intestine. So does a drug molecule get through a layer of cells? That drug needs to not only get through the cells, but the membrane that supports those cells. If you think about this, the picture of the membrane on the previous slide, it's very lossy. There's lots of loss there, and it requires larger volumes to do these assays. These assays right now are relatively low throughput. However, the early screening steps are high throughput, and this is really that issue of loss of material due to the membrane. This is how we plan on addressing the market. We already have products in the electron microscopy market. This is a research area. We have products in beta testing right now in separations as well as cell culture. We've gotten this far based on angel funding we raised 2 years ago. I'll talk about that in a little more detail in a few slides. And we're moving towards the much larger biopharma and CRO markets as we raise more money. Thank you. These areas are in purification and isolation of molecules, as well as drug permeability and drug toxicity. The specific products we'll be addressing in separations and cell culture look like this. This is basically a drop-in replacement for conventional products used in the laboratory for separations. However, we have the enhanced capability of allowing people to fractionate and actually separate their molecules, not just concentrate them. We'll be scaling that up to array-based separations. These basically drop into the same sort of robotic systems that are already used in high-throughput screening. We're doing the same approach in the cell culture space. We'll initially offer research products for cell co-culture, stem cell differentiation. We have these products in beta testing right now. We'll be scaling these up to screening and for the biopharma industry. We have a well-rounded team. Our management team consists of Rick Richmond, myself, and Chris Streamer. Rick's a serial entrepreneur, started several successful companies in the Rochester area. Before that, he was vice president of the Nalgene Group, working actually on membrane-based products for the life science industry, as well as Barnstead, the membrane-based water purification company. Chris is the technology inventor. He's still with the company. He's a recognized thin films expert, requested to be at conferences all across the world working on really thin films, and that's how we make our membrane. We are true experts in thin film technology. We also have 2 fabrication engineers who fabricate our membranes currently, a biologist, QA/QC technician, and administrator. With the funds that we're raising, we'll expand our team to include business and applications development as well as sales and marketing. Our business model is first to enter the research markets. From there, we want to enter the biopharma and CRO markets. To get into the research markets, we'll be selling separation and cell culture devices that we produce ourselves. We'll be selling these directly and then through distributors. We've already done this successfully with the microscopy product, which we offered initially basically through a web presence as well as attending technical conferences. And very quickly, we are adopted by several of the distributors. We now sell through 6 distributors internationally and through ourselves, with about half of the sales coming through Simpore and half through our distributors. We'll take the same approach with the life science products and separations and cell culture. Once we reach that adoption, we can move into the higher volume, more lucrative markets in biopharma development. We'll again produce devices ourselves. However, we'll also seek joint development agreements with larger companies that already have experience with this marketplace. Companies such as Millipore and Paul, who are membrane companies, life science tool companies such as BD, Applied Bio, as well as pharmaceutical companies themselves. We already have a number of relationships with these companies at this point. And we'll sell licensees to these companies, particularly the life science tool companies, to incorporate our membranes into their products. Funding for this will come through cash flow development as well as partnerships. Our milestones to date, we incorporated in 2007 and exclusively licensed IP from the University of Rochester, where the technology was developed. We launched our first product in 2009 based on development we had done with our angel round. We used the angel money to take the technology outside of the university and scale up production, and we're now successful at scaling that production up. Our microscopy product to date has generated about $150,000 in revenue. We expect that to continue to increase this year. We'll be launching our products in separations and cell culture. We expect about $300,000 in revenue this year. We anticipate cash flow positive the end of next year, and at that point, that's when we'll be entering the biopharma screening markets. This technology has a high gross margin. We estimate it'll reach 70%. We're actually already at 70% in our division that sells the microscopy products, with a high potential for revenue down the road. Our competition are conventional companies as well as life science device companies. However, we see these competitors as potential licensees and acquirers down the road, and that's why we've begun these conversations and relationships already. Our A-round investment is in the neighborhood of $600,000 to $1 million. We're about halfway through at this point. We just started a few weeks ago. We plan on launching our cell culture and separation products that are in beta testing. We'll scale up our production and our development efforts for our next round of products, as well as recruit business development applications specialists. And of course, provide working capital. As all of you know, the financial industry has changed, and we really need to make sure we have a cushion for that. In summary, we have a very novel, disruptive technology. It's gained a lot of interest in the membrane and device companies. It's competitively positioned against that much older technology that really hasn't changed in a number of decades. And we have experience already at reaching early adopters in the microscopy area where we'll be using that same expertise to go into separations and cell culture. And for our investors, there's a very active merger and acquisition market. Many of the device companies, membrane companies, have made many acquisitions of unique technologies like ours over the last 5 years, and it really hasn't died down in the last year. I have more information on the financials, a lot of things that I skipped over in our executive summary, which is available on our table in the back. And thank you very much for your time.